Hello, everyone. Welcome to the assembly session. In this session, we will hear about our latest and greatest in genome assembly, polishing, and curation, and many, many more. Due to the interest of time, let's jump into our first speaker's talk. Our first speaker is Jonas Korlak, who is currently serving as a chief scientific officer of Pacific Biosciences, or PacBio. Jonas has been actively involved in many scientific researches as a scientific advisor, as well as a big supporter for the VGP, Earth Biogenome Project, Darwin Tree of Life, and many, many more. As we have to start thinking to assembling and finishing the most challenging genomes, large in genome size or small in sample size with various heterozygosity and repeat levels, I asked him to share the work and progress regarding this. With that, let's welcome our first speaker, Jonas Korlak. Jonas, take it away.
Hello, everyone, and thank you, Arang, for allowing me to give you an update on what you just talked about, and specifically our assembler IPA. With 10 minutes, I unfortunately won't have the time to go into detail what the improved and phased assembly algorithm is, but I'd like to refer you to other presentations that have published. In particular, my colleague Ivan Sovic has given a talk at the Smart Leiden meeting earlier this year. We had a blog post, and then there's also information, of course, on the GitHub page. But in a 1-minute summary, the IPA assembler is a new assembler that has been optimized for the HiFi reads that you've heard a lot about, and that are also now directly output of the Sequel2E system that we announced on Monday. It features a new overlapper, a full pairwise alignment. It has a novel phasing algorithm that is de Bruijn graph based. It has its own implementation of a string graph and the consensus sequence generations using Racon. So then what you get with this is very fast, quick turnaround time for these assemblies. And that's useful because it allows you for very rapid turnaround of parameter optimizations depending on the size, complexity, heterozygosity of the genomes. It outputs high quality assemblies with high contiguity, fully phased haplotags, hyper-based quality of the polished assemblies, not needing any other technologies for generating a high quality consensus. In contrast to some other HiFi-based assemblers, no genome size estimate is needed for starting. It's relatively more conservative in the contigging and phasing, and it's very easy to use and flexible. You can either run it locally or on a distributed cluster. So the first thing Arang asked me to talk about is what's new. And a few weeks ago, we released the version 1.3. The main feature therein that it now incorporates purging of the duplicate haplotags from the primary contig set. So this currently happens to all the current assembly tools. HiFi ASM has this purging built in. And we decided to use this excellent tool developed by Deng Feng, Shane, and Richard Durbin and others at the Sanger Institute called Purge Dupes. And that's now plugged into IPA and is on by default. The second improvement is with regard to mitochondrial genome assembly. We believe that because it's circular, it should be, it's automatically excluded from and separated from the final nuclear genome assembly and then treated accordingly for a circular genome assembly. So it is, you can then identify it in the 10assemble folder, rotate the start coordinate. In the instances that we've tested, trimming overlapping ends is not needed here. So in other assemblers, because it's circular, the mitochondrial genome will be output as a concatemer. And with IPA in the 1.3, the cases that we've observed, that's not necessary. So it's already trimmed. And then we would recommend that the reads for polishing are mapped to a mapper that is aware of the circular nature. And we recommend the tool Raptor. And let me show you this here. So what Raptor can do, and you see in the lower right, the cartoon where you have a circular genome that is has a breakpoint to linearize it. Most of the reads will have mappings on both sides of that breakpoint, and Raptor is able to do that. In this IGV screenshot, you see every row represents a single HiFi read, and you see that these mappings are now giving an even coverage, and maybe not so much for consensus, but for accurate determination of heteroplasmy and structural variations, this can be quite useful. And then consensus, the same as with Rakan. So this is currently not yet fully implemented, but is available on command line. And if you're interested in this workflow, contact me and I'll share with you the scripts that are available. The second, what's new, is the implementation options. So with the SmartLink version 10.0 that we also announced on Monday, there is the option of a cloud implementation. Right now it's Amazon. And so you have automated data streaming to the cloud and no need for an internal HPC if you want to run these assemblies. And on the command line, you can also launch SnakeMake jobs into the cloud. And so then as follows, Arang asked me a number of questions that I want to address and for the last 5 minutes of my talk. The first one was the effects of heterozygosity with regard to haplotype separation. And so here, of course, the paradigm has completely flipped compared to a few years ago when it was all about inbreeding and making things as homozygous as possible. Now it's the exact opposite. So the more heterozygous the alleles are, the better it actually is for the assemblers and the better the haplotypes are being separated. And so here are just 2 examples of relatively highly heterozygous organisms, the Atlantic bluefin tuna on the left and the red admiral butterfly. And what's highlighted here are that the genome sizes for the primary assembly, the principal haplotype, and then the haplotypes are almost the same, and in this case are exactly the same. So both alleles are completely recorded and separated, and you get a 2N genome assembly. The second question with regard to heterozygosity is the question of phasing. And so there it is dependent on the level of heterozygosity as well. And on the left side is human genome, which is on the extreme end of being on the homozygous end. And so there we are dependent on the SNP density. And so that's about as good as you can do with haplotic N50s here, which are equivalent to the phase contig N50s of about 300 or 400 kilobases that we see. So that means most of the genes will be fully phased. When things are more heterozygous, you're doing much better. So this is a Drosophila F1 hybrid. And so here, in terms of contiguity, there are megabase contig N50s, megabase phase block N50s, and the phasing accuracy is extremely high because you have lots of heterozygous SNPs that are being phased with IPA. Arang also asked me, what about other programs that are being incorporated, in particular Falcon Phase, which allows you to not in addition to the haplotypes being phased, also now phase the primary assembly. And the good news here is that the file types and the concepts are exactly the same as it was before with Falcon Unzip, with the Falcon Unzip output type. And so the only thing that's needed to implement Falcon Phase for IPA involves just renaming some of the contig names in the primary from this convention to this convention, and the haplotypes from this convention to this convention. And on the right here, if you want to watch it and pause it, is some logic and some path and code on how to do that. The next topic refers to polyploid genomes. And so this, I would say, is an open area of study and is really ready to be explored. And we're seeing data and assemblies coming out that show that with the HiFi data, they're really ideal to separate these alleles. And it depends on the type of polyploidy and relative to the heterozygosity and how well these subgenomes can be either accurately separated, or you get one primary contig and multiple haplotypes. And one example was shown back earlier this year on the tetraploid rose, and showing down here, you see very clearly how nicely the HiFi reads separate the 4 different alleles, and the SNPs are being phased. And actually, just about an hour ago, Jeremy Schmutz presented at this very conference about tetraploid cotton. And if you haven't seen this in the other track this morning, you can watch it in the catch-up session, but recounting the history, and now with CCS, getting contig N50s of 71 megabases, under 100 contigs. So really showing very nicely that for polyploid genomes, the alleles can be really nicely separated with the HiFi reads. And the last question that Arang asked me was, well, what about very large genomes? So we have investigated this a little bit. IPA worked really well for the California Redwood Genome Assembly Project. And for this conference, we reran, so this project was run in March, but we reran it just a couple of weeks ago with the 1.3 version. We had 22-fold coverage. The haploid genome size is about 10 times of the human genome. And IPA plus PERCH-DUPES again worked really well, exactly on target with regard to the primary assembly size and a nice contigant 50 of over 1 megabase, and then substantial second allele and also half a megabase. And so, so this is a very large organism. We have DNA input protocols that can go down. You've heard about the mosquito protocol. A couple of weeks ago, we launched an ultra low input protocol that can get high quality genomes for really tiny species as well. And then the last point that I'd like to make, other very large genomes have been reported using other assemblers. So the other two assemblers, HiFi-ASM and HiCanoe, and I've listed them here from a talk given by Kevin Fengler, I think about 2 weeks ago at a webinar, looking at the oat genome, 11 gigabases in size, 35x coverage of HiFi data with a contigant 50 of 71 megabases. I was involved in a bird and a mammal. They also went really well. And similar sizes here, cotton and pearl millet, several gigabases, about 30 to 40x coverage. And so these are contigant 50s. Those are not scaffold N50s. So really nice results in contiguity. So I wanted to include as the last slide, some results from HiFi-ASM and HiCanoe to note that IPA is the assembler that we have developed and we support, but the other two assemblers are really outstanding assemblers. They deliver very nice results. And so if that's what you're using and that's what you're happy with, keep doing that. And I think there's good opportunities for the developers to look at the different algorithms and improve those even further. So with that, I'll close and thank our developers, Ivan, Zev, Chris, and Jim. And I'd like to thank you for your attention.
Thank you, Jonas. Thank you. Okay, let's see. We have a few questions coming in. The first question, somebody asked, have you compared IPA to HiCanoe? Did you get a chance to compare results?
Yeah, so we and others have done this, comparing IPA, HiFi-ASM, and HiCanoe. And like I said in the last slide, they all work very well. Depending on the genome, sometimes you get slightly different results. And we have seen that in some cases, IPA, HiCanoe, or HiFi-ASM works a little better than others. I outlined the differences with regard to the mitochondrial genome assembly, which is typically a concatemer and is part of the nuclear assembly. So you have to do different things in removing that and curating that. I would say, in summary, if you use one of those and are happy with it, then I would say stick with it and use that. But the other thing is that because of the HiFi data, the assemblies run now so fast that you can also run all three of them and see which one gives you the best result. And because the algorithms are quite different, I think in some aspects, for some of the genomes, it works better than others. But having said that, we have seen quite uniformly that all three give very good assemblies. So you're really looking at small differences there.
Mm-hmm. Okay, and the next question is, thank you, great talk, and asking about your experience regarding PERCH-DUPES. Have you noticed if that worked well for repeat-rich genomes, or did you have any issues regarding it?
So I'm in discussions with Dangfeng and Shane. PERCH-DUPES has worked very well overall. We have noticed that in some cases, PERCH-DUPES has a little bit of trouble finding the right cutoffs. And so then you go back and look at, and that becomes apparent in the Bosco scores that you have too many duplicates in the primary and so forth. And you can go either to the GenomeScope plot and then set the manual cutoffs, or use the HIST Python tool that's available by Dangfeng and set the thresholds manually and then rerun it, which is very fast. And then it purges quite well.
Okay, thank you again, Jonas. Let's give him a big virtual applause. Then, yeah, thank you. Okay, and then let's go to our next speaker, who is Giulio Formenti. He is currently a postdoctoral researcher at Laboratory of Neurogenetics of Language and Vertebrate Genome Laboratory at the Rockefeller University. Giulio is heading the second wave of the Vertebrate Genomes Project assemblies and contributed in establishing the first VGP pipeline and training students. And over the past summer, he worked closely to polish up the consensus with the T2T assembly, which you probably heard earlier from this conference. And while doing that, he came up with this very exciting idea that he will present, which will benefit all of our assemblies, especially those predating the HiFi. Okay, let's welcome Giulio, and Giulio, take it away.
Good day, everyone. Today I'm presenting Murphy, a tool for improving consensus accuracy during polishing via k-mer-based validation. This tool was developed in the framework of the Telomere to Telomere Consortium and the Vertebrate Genomes Project. The motivation behind the development of this tool is that assemblies contain base calling errors, and these errors can affect downstream analysis, such as gene annotation. This issue is often exacerbated in noisy long-read based assemblies, such as PacBio and ONT assemblies, and therefore polishing has become common practice for these assemblies to improve the base calling accuracy. However, one problem with polishing is that no single set of criteria can be defined on how to filter the variance using during the polishing steps. In the case of the Vertebrate Genomes Project, this can be an issue because this is the VGP standard 1.6 pipeline, and we have 4 polishing steps highlighted by the arrows here. So the first polishing step here and 3 polishing steps at the very end of the pipeline. First of all, in order to improve the polishing phase, we need a way to measure the actual base calling accuracy. So we need a way to measure the QV of the Phred score for our assembly. And one way of doing that was recently developed by Aran Ri in the tool called Mercury, which is based on measuring the k-mers that are seen in the consensus but never seen in the read set. So let's say this is your assembly, and you can count all the k-mers across the sequence. And for instance, these 2 k-mers here are never seen in the read set. So these are very likely assembly errors because they never occur in the read set. So we can then estimate the QV based on the number of errors over the total number of k-mers that in the assembly and in the read set. So for instance, if you take the sequence down here, assuming that by using this method we could identify as missing k-mers all these bases in red, this will turn into a QV of 10, so about 1 error every 10 bases. We apply this approach to the VGP assembly pipeline, and the VGP assemblies generated so far that you can see here. So here we have, at the bottom, we have the assembly pipeline steps from the early contiguing phases with Falcon throughout the assembly polishing steps up to the curated final version of the assembly. And you can see that there is a general tendency for the QV to increase over the steps. However, there is a mixed situation here in the middle where this hour polishing step sometimes produces a better QV, some other times actually decrease the QV quality. And I'm going to highlight this problem in these 3 examples where in this particular step, the QV went down. So why is this a problem? This is a problem, as I mentioned, for instance, for annotation. This is an example where in the links, this particular gene shows an indel, which is the result of having a misassembly, so a base calling error in the assembly. And we know that these kind of indel errors commonly affect 5 to 10% of coding genes in PacBio assemblies that are corrected with traditional methods. When this happens, the assembly is compromised because the coding protein gene will actually have to be corrected during the annotations phase, and will potentially introduce, like in this case, a missing amino acid. When these kind of indel errors affect more than 10% of coding genes, it's a cause for exclusion from Ref6, so your assembly is not going to be annotated. And one problem is that it's actually difficult to evaluate this problem prior to the annotation, prior to us annotating the genome. I'm crediting François Thibault, Nissen from NCBI for providing these results. So what we did was to take the K-star, a concept originally introduced by Adam Philippi in 2008, and revise it to adapt it to improve the quality of the assembly and the base calling accuracy in particular. So the K-star allows us to identify regions of the assembly that appear in an unexpected copy number. It could be either over or underrepresented compared to the read set. So in a way, it's like we're giving a third dimension to the approach that I described before with Mercury. So we now know, for instance, that if in the consensus sequence we see a particular chimer twice, but in the read set it's supported one time, then it's very likely that one of these two copies is actually an error. So we applied this principle to Murfin, and the way we use Murfin for this is that we take the set of your variants generated by the variant caller. It could be any variant caller. It could be Freebase, GATK, Pylon. It could be Aura, as in our case. It could be deep variant and so forth, or even a combination of variants from these variant callers. And what we do is we evaluate using this K-star. So let's take this example here. We have a reference sequence, and we have two variants called in this reference sequence, which we will respectively introduce a TA deletion in one case and a TA insertion in the other case. And what Murfin does is that it's going to evaluate the chimer consequences of applying these two variants to the reference. So for instance, if you apply only the first variant, we introduce this indel, but what we observe is that the number of missing chimers doesn't change. So we still have 6 missing chimers, 6 very likely errors in this reference. On the other end, if you apply the second variant, so we introduce the TA, we now remove all this, and we can also evaluate the combination of these two variants to see whether this is actually the best option. In this case, it's also fixing the reference, but the average K-star, which the closer to 0, the better, is actually only when we apply the second variant. So in this case, we would refute the first variant and only introduce the second variant. We apply this approach to the 3 cases I mentioned before. So the blue whale, the mute swan, and the leaderback sea turtle. And here you can see the results in the case of the blue whale. So this is the number of frame shifted genes. So this number, these genes that contain indels based on the NCBI annotation, which is well above 10%. So that would be a problem, a case for exclusion. And I compare this with a brute force approach that I developed earlier on before coding Murfin. And you can see that that approach was still able to correct some of the errors and bring this number of genes below 10%. So valid for annotation, but Murfin does better than that. But maybe it's more impressive when we compare the polished versus the unpolished results for other cases, like such as the moot swan or the leaderback turtle, where we don't have any brute force approach involved. And you can see here that the number of frame shifted genes goes from, in the unpolished version, from over 15%. The polished with the canonical approach, that it's part of the VGP pipeline 1.6, will only lead you to a 12% of frame shifted genes. So again, this could be excluded from annotation. But when we apply Murfin, we have that number. So we now are very well within the range that is acceptable for annotation. And the same applies to the leaderback sea turtle, where again, we have half of the frame shift genes that are removed by just applying Murfin to the variant call set. So in conclusion, the finished quality of long read assemblies is error prone. We will see with Hi-Fi, but that's at least the case for CLR and ONT-based assemblies. And these errors have a negative impact on annotation. However, we show here that Murfin can significantly improve polishing of genome assemblies. So please try Murfin whenever you have to polish your assembly to see whether you can get better results. Murfin is available on GitHub. You can see the link here, and we are preparing a manuscript to describe how it works. I would like to thank and acknowledge in particular Henri, who coded most of the principles that I described here, but also Françoise Thibault from NCBI, who helped us validating Murfin results by performing several rounds of annotations on the same assemblies. And also all the other people listed here, in particular Eric Jarvis, my mentor and PI, who contributed to the discussion. Thank you so much.
Thank you, Giulio. That was very fascinating. So I'm waiting to, let's see if we get some questions. Oh, they just got coming in. I think mine was having some trouble. Okay, so let's begin with the first one from Jeremy. How do you tolerate Murfin indeterminate Illumina reads, which occur frequently, particular in the AT kind of region in your example? I think it's asking for the AT enrichment in Illumina.
So that would be a problem. Any bias in the read set, of course, will bias also our estimate of the K-star. There are, however, methods to actually correct the bias in the Illumina read set, or potentially also the bias in the Hi-Fi, I mean, the relative small bias that there is in Hi-Fi reads. And we would like to investigate that too. So if we can improve further the estimate of the K-mer multiplicity in the read set based on a correction of the AT, GC, or whatever GA bias you may have.
Yeah.
One good thing, if I may, one good thing is that here we're not calling variance using the K-mer multiplicity. Here we're just evaluating what is the best combination, what is the best option for any particular region given the read set. So we are essentially conservative in that respect.
Great. I think we can have one more quick question. So if the concern is regarding the frame shift errors, are there any implementations with RNA-seq data directly for correction?
Honestly, I don't think we have discussed that yet. On top of my head, I would say yes, but yeah, I need to think a bit about it.
Okay, good. Thank you again, Giulio. And let's move to our next speaker, Kirsten Howe. Kirsten Howe is leading the Genome Reference Informatics team at Sanger Institute and is one of the founding members of the Genome Reference Consortium. The widely used zebrafish, human, mouse, and chicken reference had gone through all through her curation. And of course, there are many, many more assemblies I can't mention, but I want to highlight that her team are the gatekeeper before finalizing any submissions from the VGP and the Darwin Tree of Life. And I believe there are more projects. Let's welcome Kirsten and tune in to learn how to make assemblies better, much better. Kirsten, take it away.
Thanks very much for the organizers to give me an opportunity to tell you all about the things that we do to make assemblies better, and as we believe, much better. And we hope that you, at the end of the talk, you will believe me and try to do it yourself. So what we do is what we call assembly curation. And in a nutshell, we've been doing this for a lot of high throughput projects that have generated hundreds of assemblies by now, and we evaluated them all and corrected them all. And the insight from all of this is that, unfortunately, currently no automated process creates flawless assemblies. They all need some work. The remaining errors can and should be addressed, and that does not need to be time consuming. We are allocating about a week per assembly at the moment. The actions we are taking out are removing everything that doesn't belong, so artifacts and contaminants. We correct all structural errors, and we identify chromosomal units. We are then feeding back to the people who generated the assemblies and also create the software in order to improve all these processes. So over time, the process should improve further. We achieve all this through looking at all data in parallel, rather than just looking at one data type and then the next. And we actually apply hands-on changes, so it's not a fully automated process. The tools we are using are GEVAL, the Genome Evaluation Browser that we have developed and that we are producing for every assembly. It's a one-stop shop for all assembly issue identification. It visualizes the assembly issues and it offers resolutions for these issues. And what we do here is we align all available data. That's the raw data that went into the assembly, but also everything else we can find in the public space. And GEVAL then marks up and lists issues so that curators can go through and look for resolutions. What we also use are Hi-C to D maps, and they help an awful lot with reassuring us of structural integrity, and they also help identifying chromosome scale scaffolds. What we do in detail for the contamination removal is we are checking for trailing ends and remove those. We are checking for gaps that are too extensive and either reduce them or maybe completely remove affected context. We check for organelle sequence, and when we find it, we separate it whilst carefully preserving numides and nupides. And we, of course, check for any sequence that is not from the targeted species. This might come from lab or data mix-ups, but it might also come from symbions or generally any cobiont that was present in the sample during the sequencing process. This is all automated, but we have someone looking through it in order to make sure that everything is as it should be. What we then address, and this is unfortunately a very, very common issue with assemblies, are duplication and collapses. They can easily be spotted in our GEVAL browser, but they can also easily be spotted in the Hi-C to D maps. And they lead to distorted biological content in the assembly by, for instance, duplicating genes or removing paralogs. So they really should be addressed. And then the meat of the curation process is addressing misjoins and missed joins. So things that were joined together but shouldn't have been, we break, and things that should have been joined together but weren't, we join. We also unscramble scaffolds that have been put together wrongly in order to assure that they are that they have every sequence in the order that it should appear. And finally, we identify chromosomal scale scaffolds and we name them, thereby also trying to identify sex chromosomes where possible and reconciling with the karyotype if known. This is sort of a tour de force through all of this, and there are many details to this process. If you are interested in this, I recommend you look at the posters by Joanna Collins, who tells you more about the Hi-C to D map patterns that you can encounter during assembly curation, and also at Sarah Palin's poster about identifying sex chromosomes across varying species. When you look at what we achieve with curation, so far we've curated 172 assemblies as of last week, and that amounts to nearly 250 gigabase pairs that we've curated. On average, we intervened 243 times per gigabase pair, and this would be breaks, joins, and removal of false duplications. And this leads to an average reduction of assembly length of about 2%, and an average increase of scaffold N50 by nearly 60%, whilst the scaffold count usually goes down by about 30%. We also achieve an assignment of sequence to chromosomes by 98%. So nearly everything really is in chromosome scale scaffold at the end of this process. And you can see here on the right where we plotted the assembly length of different vertebrate clades, clade assemblies, against the number of manual interventions per gigabase pair, that every assembly needed interventions and that the number of interventions is very varied. So the lowest number, if I remember correctly, was 4 corrections to an assembly, and the highest number went into the thousands, several thousand. If you want to find out more, even more about this process, I would like to point you at a manuscript that we've recently uploaded to bioRxiv. It's called Significantly Improving the Quality of Genome Assemblies Through Curation, and it details the whole process and it makes recommendations of what you can do if you don't have the Genome Evaluation Browser present. Also, recently, Dangfang Guan has set out to replicate the whole analysis process of analyzing all data in parallel in an automated fashion. And the tool he developed is called Asset, and you can find it on GitHub and hopefully also soon in publication. I don't want to leave you without pointing at the assembly standards that the Earth Biogenome Project has defined, because by performing the curation, as I talked about just now, you actually comply with those standards. So the EBP asks that all assemblies have assured quality by containing only standard bases and no trailing ends, that they are decontaminated and that all organelles are separated. Ideally, haplotypes should be separated as well. Then all assemblies should have gone through processes of identifying all the discordances between raw data and the assembly in order to locate and remove structural errors. If the karyotype is known, you should always try to reconciliate it with the assemblies you have produced and identify sex chromosomes where this is possible. In terms of assembly measurement, the EBP recommends only to release assemblies that have a sequence quality of at least Q40 and a continuity of at least 1 megabase pair in terms of contig and 50. Of course, where this is possible, there might be assembly structures where this will never be possible due to the chromosome size of the species. They also recommend that you attend chromosome scale scaffolding, and this should reach at least 90% of the sequence. Less than 5% false duplications should remain, and the KMA completeness should reach at least 90%, as well as the gene completeness, measured in, for instance, WUSCO single copy genes that should reach 90% as well. Also, they expect to have 90% of transcripts from the same organism to map to the assembly that was produced. The EBP also recommends to follow a certain sample naming scheme that was pioneered by the Vertebrate Genomes Project and the Darwin Tree of Life project. This assigns every sample a unique name that consists of 4 parts. The first is a 2-letter clade-specific prefix, then a 3-letter genus-specific abbreviation, then 4 letters of the species name, followed by a number for the individual that was sampled. And here on the right, you see an example. Il-al-Crepa is the butterfly Alcis repondata, and it's the first individual that was sampled of this species. This allows to identify the taxonomy context of the sample that we're talking about. So for instance, if it has the prefix I know it's an insect and not a plant or something else. And it facilitates the recognition and also, as we've learned, the communication about samples and assemblies. If you then produce an assembly from a sample like this, we recommend that you append an assembly version number. For instance, make this Il-al-Crepa 1.1 or Il-al-Crepa 1v1 or something like this. All the rules, details, and existing lists of all entries in GenBank, or species entries in GenBank, can be found at the URL down here. If you have any questions about this, please just email me. We are trying to set up a repository that allows to register names for samples. And last not least, the EBP also recommends a certain submission structure for your assembly. And if you start at the bottom of the slide, your primary assembly, alternate assembly, so the other haplotype or an alternate variant, any cobiont assembly that you identified, and of course all the raw data should all be submitted under their individual data bioprojects, and these all should be gathered in an umbrella bioproject that is specific to the species. This umbrella project can then be superseded by another umbrella that is specific to your project, but should also be attached to the already existing Earth Biogenome bioproject, so that all the assemblies that have been submitted can be easily identified from this one Earth Biogenome Project umbrella bioproject. And these are our recommendations for today. Thank you very much. And I won't leave you without showing you all the people that work on assembly curation. This is the Genome Reference Informatics team at the Sanger Institute. And thank you very much for listening.
Thank you, Kirsten. That was fascinating. Okay, we have several questions. I guess the first one is related to the availability or reproducibility. GEVAL seems essential for all assembled genomes. Can the users upload their own data for curation? Or is there something available like a portable Docker kind of to the research community?
So unfortunately, GEVAL and its production are really tied into the Sanger infrastructure. So this is currently not portable. We've tried to come up with recommendations in order to make curation possible without GEVAL. And I've described that in the manuscript that we uploaded to bioRxiv. So I hope that will assist people in going through similar curation processes as we go through as well.
Okay, the questions are just coming in. The next one is your assembly size versus the correction plot shows an exception, large correction rate for fish. Is that an artifactual or biological thing?
I think it's biological. Fish are known to be nasty amongst people who create assemblies, at least certain fish, not all of them, obviously. There seems to be an issue with heterozygosity and repeat content that creates things that need to be corrected by hand because the current assembly processes can't quite get it right. But even there, I think we've made serious advances with new assembly software and also with new data types that have been available. So some of these data are somewhat older, let's put it that way.
Thank you very much again. People really thank you for, yeah, somebody says, good to know the evaluation process and requirements. I hope it helps to improve their assemblies as well. Okay, our next speakers, we will have two short talks by Zemin Ning and Jay Gure. Zemin Ning, we start with Zemin Ning. He is leading the high performance algorithm group at Sanger Institute and has been actively developing methods regarding genome assembly and scaffolding tools. His previous scaffolding tool for linked reads, namely SCAF10X, were chosen as part of the VGP assembly pipeline, and pretext series is widely used for Hi-C-based curation. And today he will share his scaffolding pipeline using Hi-C, I believe. Zemin, take it away.
Hi, everyone. Today, I'm going to talk about a new Hi-C scaffolding pipeline, which is an integration of a number of existing assembly packages. In the community, there are a number of Hi-C scaffolding tools developed by different people, different groups, algorithms different, implementation different, of course, performances are different. Recently, my colleague Kirsten Hall and her team in the Tree of Life project have evaluated 3 packages, 3DNA, SOSA, and SCAF Hi-C. So let's say for this 940 megabase mouse genome, 3DNA did a good job in scaffolding. You can see here chromosome scale assemblies and better than SOSA and SCAF Hi-C. However, at the context level, it is fragmented. The context number is 1,264, larger than 305 from SOSA and 304 from SCAF Hi-C. So by looking at the details, they found actually the contexts are correct. There's no need for such breaking. To solve this problem, we developed a new pipeline, which integrating a number of existing packages like Highline, Break Hi-C, 3DNA, Course, SCAF Hi-C. The Highline procedure mainly deals with data processing, filtering, and alignment Hi-C reads. At the end, you will have a sorted bound file. The Break Hi-C identifies context errors and remove errors at the context level. 3D-DNA does genome scaffolding to get chromosome level structures. CORS stands for chromosome assignment using synteny. It uses the context from break-Hi-C to replace sequence in the 3D DNA assembly. And finally, scaf-Hi-C, it removes errors in the scaffolds and makes alternative joins with broken context. The CORS is a key stage in the pipeline, so I'll highlight some details here. So we have a bunch of contexts. For each context, we break into small fragments, let's say 1 kb or 2 kb. As there are repetitive elements there, you can see scattering everywhere. So those fragments need to be removed. By doing that, we can uniquely place each context in a place and build up a new structure, as shown here. So we have obtained a new chromosome. Back to the Morse maps here, as you can see, the ladder pipeline produces same scaffolds as those did by 3DNA. As you see here, the context number 304 is similar to scaf-Hi-C and SOSA. Another example for the butterfly genome, you can see here, ladder produce very nice scaffolds here. The context is low 126, similar to scaf-Hi-C and SOSA. You might find here there is an error in the scaffolds and actually has been corrected by the new pipeline. We also have done the human genome sampling, as you can show here. So the context number has dropped from 2,700 to 2,200. And also you can see here for the scaffolding actually has been improved by the new pipeline. The number of people involved in the project, I want to thank Ed, Kirsten, Joe, Marcella, Annan, Shane, and lastly, Richard Durbin. Also, thank you very much for the audience.
Thank you, Zemin. As we are waiting for questions, I guess we can jump to the next speaker and then take the Q&A all together. So our next speaker is Jay Gray, who is a senior bioinformatics scientist at Dovetail Genomics. When Jay was a graduate student, he developed SALSA that you just heard from Zemin's talk before, which was part of the initial VGP pipeline. And today he's going to share that Hi-C can be used not only for scaffolding and also for phasing. Let's hear more about that. Take it away, Jay.
Hi, I'm Jay Gray from Dovetail Genomics, and in this talk, I'm going to describe some of the recent developments with OmniC data to get chromosome scale haplotype assemblies with PacBIOS Hi-Fi reads. As you might know, Dovetail Genomics has a new Hi-C protocol called OmniC that gives you the uniform sequencing coverage along with the traditional long-range linking information obtained by the Hi-C data. So I'm showing the coverage plots here for 3 different Hi-C sequencing technologies. So the first one is the one with single restriction enzyme. And you can see that the coverage is not at all uniform. The black dotted line here shows the expected coverage that you would get with the shotgun reads. And Hi-C with single restriction enzyme is very far from that. If you upgrade slightly and use the multiple restriction enzyme Hi-C, the coverage slightly becomes uniform, but it's not at all close to the one that we would expect. However, if you use OmniC, the coverage is much more uniform compared to any of the traditional Hi-C protocols, and it matches close to the expected shotgun sequencing coverage. So we can leverage this property of OmniC for a variety of different applications, and one of them I'm going to discuss here today. If you recall from last year, this preprint from Shilpa Garg and her group came out to generate phase assemblies with PacBIOS CCS and OmniC data. So in our work, we were inspired from that, and we slightly modified that pipeline to use OmniC data for chromosome scale assembly. So the high-level pipeline is as follows. You begin with PacBIOS CCS reads. You assemble them with any choice of assembler. In this case, we use Peregrine. Then you scaffold those with OmniC to get chromosome scale scaffolds. In our case, we used HiRISE, which is Dovetail's own Hi-C scaffolder. Then you align OmniC reads to those scaffolds and call SNPs using FreeBase. Then you phase those SNPs using HapCut2, and you could do chromosome scale phasing because OmniC has those long range links that can give you the chromosome size phase blocks. Then you align CCS reads back to this chromosome scale scaffolds and look for the heterozygous sites where you can get evidence for reads phase. So essentially, at the heterozygous site, based on the allele present in the read, you could either put them in the haplotype 1 or the haplotype 2 piles. And once you have these reads bent, you could assemble each haplotype independently, and you can further scaffold them with phase-specific OmniC reads to finally get the deployed phase assembly. We ran this pipeline on a couple of human genomes. So I'm showing here the results for NA12878. You can see that after running this pipeline, both the phases have 148 megabase N50, which is close to chromosome scale assembly. And down here, I'm showing the contact maps for 2 haplotypes. And you can see that each chromosome is there in its own scaffold. And there are very few cross-chromosomal Hi-C contacts, implying that the scaffolding went extremely well. So this results speak for the continuity, but if you want to assess the correctness of the phasing, we could look at the parental KMER markers. So these markers were derived from a TrioCanu project where they had data for Illumina data for parents. And here what we wanted to check is how unique is each contig in a particular haplotype for either maternal or paternal KMER markers. So each dot here is a contig, and on X and Y axis, we are plotting the counts of maternal and paternal KMER markers. So if phasing works okay, then most of these points should be clustered around the axis because they would have abundance of one of the parental KMER markers. And that's what we observe here for NA12878. Most of the contigs are well phased except these 2. We've dug down further on what's going on, and we came to the conclusion that some errors in the scaffolding had caused errors in the phasing, and that has further propagated these errors in the downstream process. Similar results were observed for HG2 as well, but the data here was much better than for NA12878. That's why we see that the contigs are very well separated in terms of their phase accuracy. And again, some of these errors are due to the scaffolding errors, but overall it looks great. So humans are easy. We wanted to test this pipeline on some non-human genomes. So we used data from Atlantic bluefin tuna. This data was generated in a project that's a collaboration between Dovetail, PacBio, and Barbara Bock's group at Stanford. So we begin with CCS assembly of bluefin tuna genome. The assembly itself is pretty darn good. It's around 26 megabase N50. After initial round of HiRISE scaffolding, the N50 became 34 megabase. Then we ran this whole deployed pipeline on those scaffolds, and we ended up with 2 haplotypes where the N50 was 27 and 28 megabase respectively. And we know that this is chromosome scale because the expected number of chromosomes in tuna genome is 24, and majority of the scaffolds are contained, sequence length is contained in top 24 scaffolds by length. In this case, we didn't know parents of this tuna, so we can't really assess the accuracy of haplotype phasing. But what we can do here instead is look for unique KMERs in both the haplotypes. So these are the KMER spectrum plots. On the left, it's all the reads which are not binned by haplotype. And this purple plot here shows that those KMERs are duplicated. And once you run them through the phasing pipeline, you can see that all those duplicated KMERs have become unique. And there are still some duplicated KMERs which may belong to the homologous regions in the tuna genome. So this pipeline works well even on the non-human genomes. And internally, we are testing it further on some of the plants. That's it for me. If you want to access this OmniC data, we could go to this URL. We have put the data for humans out there already. The data for tuna is submitted to SRA, but we will release it after the paper has been published. And if you have any questions, let me know. Thanks.
Thank you, Jay. Unfortunately, Jay cannot be with us in person, but we have Thomas Swale. Is that correct? Yeah, we have Thomas Swale to answer questions regarding to his talk. Okay. So while listening into Jay's talk, we got a question to Zemin, and also I have a question myself. The first, is Ladder available to the users?
Thank you. It's not, as a whole pipeline is not ready yet. We are working on it. If people are interested, can contact us maybe, because individual modules are available now. So contact us, we might help. But in the long run, of course, we will release the Ladder as a whole pipeline.
Mm-hmm. And my question was, if there were any remaining alternate haplotics that should have been removed in advance, but somehow were incorporated, will they be recognized and handled, or would you rely on the former process?
We at the moment we are relying on Kirsten's team to check haptics or any other downstream problems. My colleague is developing image-based scaffolding tools that might be help in the future.
That sounds very promising. Yeah, and I guess that another question, are you preparing Ladder to be applicable to graph-based assemblies that will be available soon, I guess, in the future?
Yeah, that's the plan. Yeah. Not graph.
Okay, thank you very much.
Yeah.
Okay, and another, yeah, and I have a question for Dovetail's group, I guess, that from the method that Jay had presented. First, have you tested the heterozygosity level of the genome that you worked on? And was there some sort of a limit on the level of heterozygosity that will work on? Because I assume that your method is based on a squashed assembly that generates one haplotype that has a sort of a mixture of both haplotypes and phase it at the end.
Yeah, it's an excellent question. So yes, process-wise, we first generate a pseudohaploid assembly. In this case, we were using Peregrine. And then from that, in concert with the OmniSeq data, we'll call SNPs, since the OmniSeq data is fairly consistent with what shotgun data would be as far as representing the SNPs across the entire genome. And then we use those two, the pseudohaploid assembly in concert with those SNP calls to parse the hi-fi reads, and then assembling those separately. So as Jonas kind of mentioned earlier, heterozygosity is kind of your friend in this instance, since it's going to make it easier to parse the reads out. We haven't explicitly tested that many cases of this yet, but fundamentally in a higher heterozygosity or higher heterozygous organism would make it much easier for this approach, since it would be easier to parse the reads out.
Mm-hmm. Yeah, and there is another question. Would OmniSeq phasing work on CLR data, or is that only available for hi-fi reads? Yeah,
so we tested that too. It's a good question. It does work, but it's an accuracy issue. So the CLR, you will have SNPs, but there's less confidence in the SNPs since the reads aren't as accurate. So you can call them, but you got to take it with a grain of salt compared to the hi-fi accuracy. So there will be some doubt in some of the SNP calls.
Okay. We have one question from Jeremy. Can you comment on the proportion of longer Hi-C links in OmniSeq versus single cut site libraries? What is the change in link size distribution?
Yeah, so it's the major difference is that digestion step using DNase instead of restriction enzymes. So they're all getting fixed around the histones to digest down to nucleosomes. So apples to apples, comparing long range links for the same organism between the two platforms, we haven't seen that much of a difference. There might be some niche cases. It's probably going to be species specific or architecture specific, but we haven't seen a significant change across the same organism with the two different techniques.
Thank you very much. I guess we are at the top of the hour. Thank you again, and a big applause to all of our speakers. Thank you very much for offering this great talks. And thank you all to the listeners. Hope to see you again.